Ready to go?
Okay, it seems like time to get started for the next session. My name is Sue Pierce, and I'll chair this session. Our first speaker is Dorian McGarven, who is a senior investigator at the NIH, a colleague of mine. And to let it be known that Dorian was absolutely instrumental in helping my lab get going on learning the pathogenesis of cerebral malaria through his knowledge of neuroimmunology. So it's been a very productive, enjoyable collaboration. But for today, the title of Dorian's talk is CD8 positive T cells trigger aberrant cerebrovascular calcium dynamics during cerebral malaria. So welcome here.
Well, thank you for that kind introduction, Sue. And really, it's wonderful to be here this afternoon presenting our work to all of you. Thank you so much to the organizers for putting this event together. My talk is going to focus a little bit on a different side of the equation here. Many of the talks focusing on the parasite. Mine is going to focus more on the immune reaction to that parasite and how that triggers some trouble in the central nervous system compartment. So that's the title of the talk. I won't say it again. But my laboratory tends to focus a lot on how infections challenge the CNS. So we focus on fungi, viruses, bacteria, parasites, basically things that interrupt what I call the barrier system in the CNS. So one of the most important barrier systems is the lining of the brain or the meninges. There's a lot of immunology going on in that compartment, and we study that sort of every day of our lives. Another really important barrier is the blood-brain barrier. And this is another system that has to be kept intact. And when it gets broken by a parasite or the immune system, that causes, obviously, complications in the CNS. Other barriers throughout the CNS, there's one that's sort of deep in the ventricular system. Our choroid plexus produces our cerebral spinal fluid. Pathogens gain access to that compartment and can trigger complications. SARS-CoV-2 is a good example of that. So disruptions of these barriers is something that contributes very significantly to CNS complications. And then the resolution of those barriers is what sort of puts us back to homeostasis. So how do we get back to health again? You have to see first how the barrier breaks down, and then you need to see how it gets fixed. And so that's something we focus on a lot in my laboratory here today. So I'm not going to go into the complexities of the meninges because that's not really the focus of my talk. I really only want to say that as the immune system starts to engage the different levels or layers of the CNS barrier system, this is all the way from the dura, the arachnoid, the pia mater, all the way down into the brain parenchyma. As vessels come down and enter this space, obviously they have a very elaborate barrier system. And this is a place where Plasmodium really causes some trouble. And it's not in every individual that the trouble is caused, but it is certainly a complication that can be fatal. And this is a complication that can affect a lot of lives. And so through the sort of intervening slides, when I put a title up, you're going to see as a backdrop, these are images from the James Webb telescope. So if you don't like the immunology and prefer the parasite, then you could just focus on the astrology here or astronomy and think about the stars. The focus of my talk today is going to be on this disease, and I really don't think it needs any introduction, so I'm not going to go through all of these points. Rather to say that it is a very serious disorder, primarily affecting children or predominantly affecting children that get malaria, has a high mortality rate. And obviously, cerebral malaria can create these disruptions of the barrier system. So, I mean, when I look at an image like that, where you see all of these blood cells around a vessel in the parenchyma, I say to myself, the CNS barrier has been broken very significantly. And the question is, how does that occur? How do we get from an intact blood-brain barrier to a situation where we have red blood cells in our parenchymal space and to have all these petechial hemorrhages throughout the CNS compartment? And so there was a paper that was published several years ago by Terry Taylor's group. It was in the New England Journal of Medicine, and it showed that in children there was very significant evidence of brain swelling, and that seemed to be the cause of death in these individuals. And so when the brain swells, you're in a closed space that puts pressure down on various vital structures like your brain stem. That can create problems very quickly, and that's called herniation, when the brain gets pushed into, for example, the spinal cord, and then that compression of the brain tissue can trigger loss of very important vital functions like respiration, your heart rate or heart beating. And so swelling, I study swelling for a living in stroke and other types of ischemia, traumatic brain injury, viral meningitis. Swelling is a very, very bad complication to have in the CNS or brain compartment. And this is just a few images taken from that particular paper. Again, you don't need to be a neuroradiologist to figure out the difference between a brain that looks relatively normal to one that is completely distended and swollen, and to see that kind of pressure being placed. And this is the brainstem here, where you're getting compression on that, and that is going to cause an almost immediate fatality. And so, surgeons are often very quick to try to relieve pressure. If you have a situation where you have fluid building up in your brain, basically they're going to try to relieve that pressure as quickly as possible to ensure that you stay alive. And if you don't relieve that pressure, there's nowhere for it to go, and then sort of that's the end of life at that particular point. So one of the pathogens that we study in the lab there is Plasmodium burgii, the ANCA strain. We basically infect red blood cells, put that into circulation to sort of bypass that mosquito phase of things. And when you put that into rodents, you see a very devastating disease that mirrors the type of pathology that one sees in humans. So you'll see petechial hemorrhages here at day 6 post-infection. This is Evans blue. It looks like sort of a Smurf brain here, all bright blue. That's a dye that's connected to albumin, should be in circulation, but should not cross the blood-brain barrier. This is what it's normally supposed to look like. And then when you put that dye in when an animal is succumbing to cerebral malaria, again, you can see this massive extravasation. You can also see hemorrhage here in the olfactory bulbs. The bulbs take a very serious hit during this disease, and that creates a loss of sense of smell. The other thing that happens is that there's such a buildup in pressure, and this is much the same as what one sees in children with cerebral malaria, that you actually get compression of different structures. So there's damage here in the olfactory bulbs, and there's also damage here down in the mouse's brainstem. And this is basically a vital dye called propydium iodide, and that's the red dots there. And then Evans blue is that white that you're seeing there. I just want to make the point that basically there's leakage in different portions of the brain, but critically speaking, when this area gets damaged or there's too much swelling, then that actually creates herniation of that structure, and then the animal will succumb to that. And this is an MRI scan from a mouse that has this disease. This was not captured by my lab, but another lab back in 2005. And you can just see the displacement of these sort of white lines. So this is being, number 1 here is being extended, and then 2, 3 in the back, you can see there's compression there. This brain is heavily swollen and is being physically distorted by the amount of swelling that's in that particular structure. So now I'm going to take you into the disease process and show you sort of how it all plays out. So what drives that type of fatal pathology in that particular compartment? And so starting with the parasite distribution, we know parasitized red blood cells can stick to the walls of cerebrovasculature, more so in humans than in rodents, but it still occurs in rodents to some degree. You're going to see some fancy movies throughout my talk today. These are captured by a 2-photon microscope through a thin skull bone. So we thin the bone down to about 20 microns, just a tiny little area like the sort of the tip of a pencil. And then we just sort of peer through into the underlying brain tissue and have a look to see what's going on. So in this film, you're watching monocytes try to pull some red, see the little red dots there? That's parasitized red blood cells that are being pulled off of the wall of the vasculature. So this is a blood vessel in the animal's brain. This is day 6 post-infection. This animal is going to succumb to this disease, but these monocytes are making a desperate attempt to try to pull the parasitized red blood cells off the wall. In this next film, you can see a parasite in a red blood cell here that is extravasated in red. And these are perivascular macrophages in the space. So these are now in brain tissue. And what they're doing is they're just jumping on top of this, this parasite. So this guy never knew what hit it. And these macrophages are going to take that material and they're going to pick it up. And then they're going to present the antigen to incoming immune cells. So you have a parasite on two sides of the wall here. You have it on the lumen, and then you have it on the ablumen. And so that's going to create an immunological environment for these T cells and other immune cells to survey. So this is what brings us to the sort of the root cause of this, which is actually the CD8 T cell. So a CD8 T cell you typically think of going out and fighting a virus, right? Inside of a cell, you sense viral peptides, and then you react to that cell and you kill it. But in this case, you have a really weird scenario that you have a parasite that's out in the red blood cell world, but it actually is going to be released and taken up by a cell, which is then going to present that antigen to the CD8 T cell. So we know in rodents very clearly from a number of studies, again, not from my group, but from many other groups, that have shown that CD8 T cells are absolutely required for this disease. So if we get rid of CD8 T cells, you can see 100% of the animals will go on and survive. That's the green curve there. If we get rid of CD4 cells, we have absolutely no impact on the disease whatsoever. And you go from all pathology, 100% pathology, to no pathology, where we just simply take away the CD8 T cell. So that told us that the cells are actually incredibly important in driving this particular disease process. So we decided to go back to our 2-photon microscope and again have a look underneath the hood of the car here and see what's going on inside of the system. And this was what I call one of those smoking gun films. You know, when you first see a film like this, you say that's the disease, right? That is the root cause of this particular disorder. So here we're looking in a symptomatic mouse at day 6 compared to the ear of that same animal. You see no CD8 T cells that are specific to the parasite in the ear vasculature, but you can see all these T cells here rounded up on the wall of the blood vessels of these mice with cerebral malaria. And you can see a number of these cells are actually static, right? So I can't use my laser pointer here with the movie, but a number of the cells are rounded up and they're physically engaging the wall. So they're stuck to it on the lumen, they're under shear flow. That means that they have adhesive forces that drive them to interact with that wall. There's also cells that are extravascular. And again, this told us, at least me, that this was probably one of the root causes of this particular disease process. So there's been some controversy as to whether CD8 T cells are similarly engaging the walls of humans and children that have this particular disease. So Sue Pierce and I, we've collaborated on this particular project that was published a few years back. Showing in children, again, these are samples that we obtained from children's brains in Malawi, comparing the patients that have cerebral malaria and high sequestration to low sequestration to these are the controls on the right. We basically were able to observe a significant number of CD8 T cells here in green adhering, this is a blood vessel here in this children's brain, adhering to the wall and engaging it, much the same as the pattern that we observe in humans. So you combine the CD8 localization plus the herniation and the swelling, they look nearly identical between these two diseases, in the children's brains and in the mice that have this particular disease. So we are, at this point fairly comfortable with at least the idea that CD8 T cells are contributing to at least a subset of these patients' disorder. And so now we came to, okay, let's just have a look at what's going on in terms of blood-brain barrier dynamics. And so this is the actual blood-brain barrier in sort of real time. So this is a calcium indicator dye in a mouse, and you're looking at the normal fluctuations of calcium that sort of spread along endothelial cells. So they're actually connected by connexins, and they sort of spread calcium from one cell to the next. And it kind of gives this oscillatory pattern, right? Sort of waves are passing along the vessel wall. And this is what things look like when everything is normal in the CNS compartment. However, during cerebral malaria, we noticed this extremely bright and intense calcium activity. This was another sign that the blood-brain barrier was deeply distressed. You can also see there's a vessel that sort of flashes like a lightning bolt at the bottom of the screen there. We would see these really intense wave propagations that would occur as CD8 T cells would, as the vessels would spread their calcium along the wall. So this film is just sort of drastically different from what I showed you under homeostasis. And so we started to go in and just quantify these interactions. So we said it's not enough to just say the CD8 T cells stick to the wall. We want to show sort of definitively that they can cause disruptions, and in this case, calcium is an indicator of that disruption using different types of microscopy. So we started measuring a baseline activity, which is shown here on the left graph. And then we started measuring the activity of the endothelial cells in response to cerebral malaria. So we're measuring dynamic ranges, which is the total range of the calcium. And then we're measuring deltas, which are the amount of calcium flux you have off of a baseline. So the delta is sort of a standard measure that we use to assess what's going on. So you come in, you start looking at arteries and veins in these animals with cerebral malaria, and lo and behold, you can find that there's really intense calcium delta activity. So lots of fluctuations in these vessels. There on the right, the red plots versus the naive animals on the left. So both arteries and veins are taking a hit during this disorder. So this gets us to the next experiment where we're going to actually go in and try to assess whether the CD8 T cell is a trigger of that event. So we have this fancy mouse where you can actually see when a T cell engages peptide MHC on its surface. So when a T cell comes up and recognizes a peptide, it's going to actually flash calcium. The T cell will do that. And then when the vessel is distressed, it will flash calcium. So we have a calcium indicator in the T cell, and we have a calcium indicator in the vessel wall. So I hope that makes sense, and that sets the stage for the next film here. So basically, these are CD8 T cells in red, and they have a calcium indicator in them in green. And then the vessel wall is also going to flash calcium, and that's where those sort of dotted or the arrowheads are shown there. So this is a chaotic mess, but you can see the CD8 T cell engages, flashes calcium, and then you see the wall flash calcium right after it. So boom, flashes, and then the wall flashes calcium exactly after it senses peptide MHC. So it's coming up, it's seeing parasite antigen displayed on the wall, flashes, and then the wall flashes in response to that. And so there is a direct relationship shown here between the T cell and that actual calcium flux on the wall itself. And so if we get rid of T cells, as one might expect, you can basically eliminate all calcium activity, aberrant activity along the wall itself. So T cell depleted is on the right-hand side, and on the left-hand side, you have the control. And you can see we have completely shut down these undulations in this particular, in the wall itself. And so then you say to yourself, okay, so how can we manipulate this? And so I'll tell you two pieces of evidence to support that the wall is driving, the T cells are driving activity along the wall. And first will be genetic, and then the second will be a therapy, right? So how do we fix this using the knowledge that we've gained? And so here, in this phase of the talk, basically what we're using is a vector that can only target cerebrovasculature. So this adeno-associated virus has CRE in it, and when we put it into a mouse, it won't go in every blood vessel. It'll only go in the endothelial cells that are comprising the blood-brain barrier. So that's the red here, and you can see this red. These are all endothelial cells and vessels here in the mouse's brain. And we put this into a mouse, wait 2 weeks, and it can target basically every single vessel in the mouse's brain, which is pretty cool to not touch any vessel throughout the body, but only touch the vessels that are in the CNS compartment. And you say, why do we want that? And the reason is we want to manipulate the wall. We just want to touch cerebrovascular endothelial cells. So here's our AAV in circulation. We put it in, comes into a cell. It has CRE, and then we have a mouse that has a FLOX gene. In this case, it's beta-2 microglobulin, and that's the light chain for MHC class 1. So CD8 T cells would normally come and engage a peptide right here and then trigger activity in this endothelial cell. So by putting in this vector, we can delete that molecule there, and then we can knock MHC off the cell surface. So without touching MHC anywhere else in the body, we'll just touch it on the wall and ask if that actually changes the disease. And so this is a really cool experiment where we were able to show by eliminating MHC on the wall, getting rid of the target for the CD8 T cells, we basically preserve about 80% of the mice in this case, which is pretty darn good that we're able to essentially create that much survival by just interfering with the wall itself. And you can see here that we get rid of MHC molecule, not to the same level as a knockout mouse, but pretty darn close to getting down to background level. So we've deleted MHC from the wall itself. So that tells us that the peptide has to be presented on the endothelial cell so that the CD8 can recognize it to drive this particular disease. And so what signaling needs to happen on the wall to get all of this revved up? And so we pulled out the endothelial cells. We did a fancy microarray, and that was well before single cell was a big thing. So we were doing microarrays at this particular time. And we said, what machinery is activated in those endothelial cells when these mice are succumbing to this disease? And the big winner here at the top of the list was type 1 and type 2 interferon. So interferon alpha beta and interferon gamma. And they were driving just about everything you could imagine. The MHC genes, the proteasome, all kinds of things downstream in these endothelial cells, including MHC class 1. So we went through and used our fancy genetic tool and we eliminated this one, then that one, and then this downstream signaling that both of these guys need, which is STAT1 here. And long story short, if you take out type 1 interferon, no impact on disease. Take out type 2 interferon, no impact on disease. But if you take out the common STAT1, then you actually stop the disease. So here, this is about a 60% survival by eliminating the common signaling element for type 1 and type 2 interferon. And what that says is that they're probably redundant, right? Type 1 and type 2 can both do relatively similar things in these vascular endothelial cells, and it can activate them to drive this particular disease. So that tells us a lot about what's going on. I said the CD8 engages, it triggers the calcium influx. Those T cells are important in that disease. MHC, the other side of it, needs to be recognized, and type 1 and type 2 interferons likely activate that endothelium to get to that state. So the final piece, and then I'll conclude, is the therapeutic intervention. So how do we fix this particular disorder? There's a couple of different options. One of these is what Sue has been working on for many years, published a really nice study here in PNES showing that you can put in a glutamine sort of antagonist here. They call it DAWN, and that essentially might starve the T cells of their sort of activities. It almost might also protect the blood-brain barrier, but this is something that's in a clinical trial right now in Malawi. So this is a bench to bedside going from this model directly into human beings. Two things that I'll tell you about here are two other things that are not in clinical trial, but they're certainly ways to stop this disease. One is to take away the effector molecule that the CD8 T cell uses. So perforin is one of those effector molecules that a T cell releases onto its target. And if you take a drug against perforin and you put it in early enough in a pre-symptomatic mouse here at day 4, you can essentially preserve 100% of those animals. So the perforin is likely popping holes in the endothelium and then dysregulating that sort of calcium, right? You're getting lots of calcium rushing in, and that's probably creating that disequilibrium. So it's really amazing here. This is I think one of the best pictures I have of this. This is the animal without the perforin inhibitor, and this is the olfactory bulb. And you can see it's just lit up with this dye that's leaking in, showing that the blood-brain barrier is broken. Here with the drug, you can see that the dye is basically not extravasating. So you get in there early enough and you're actually able to stop this particular disease. The downside is if you wait too long, so if you wait to the symptomatic phase and start around day 5, you basically cannot stop this disease. And so once there's a critical threshold that is crossed, then this drug, this inhibitor is no longer able to achieve its goals, right? So that's another thing about thinking about therapeutic intervention is you have to get there quick enough. And the last piece I'll leave you with is something more crude, but I liked it because it was just a way of knocking those T cells off the vessel wall. So think of the T cells, they're stuck to the wall, much the same as the red blood cell is. They're engaging, releasing effector molecules, and I just want to get them off there, right? Not block perforin, but literally just get the cells off the wall. So we decided to come in with sort of two drugs that were previously FDA approved. This is a VLA4 inhibitor and an LFA1 inhibitor. Those are adhesion molecules that T cells and other immune cells use to stick to the vasculature. The VLA4 drug is called natalizumab. You might know that from treatment of MS patients. And we decided to go in this sort of mid-symptomatic phase here at around day 5 and apply just for this period of time. And when we did that, we promoted about 75% survival in the rodents. And I just want to show you what that looks like. This is a movie of that. So pre-treatment, the T cells are sticking to the wall, right, nicely adherent. And then post-treatment, boom, they just knocked them right off the wall. It's an amazing thing that just those adhesion molecules were enough to keep them physically tethered to the lumen of the vasculature. But doing that, again, at that symptomatic phase was sufficient to save the mice of this particular disease. And this is just the quantification. I think the movie speaks for itself. This is before adherence, and then sort of after the treatment, you can see it just knocks them right off the wall. So that's all I have in terms of my themes here today. In terms of people that contributed to this particular project, there are many. Bo-Ran Choi is a postdoc in the lab. She's doing a lot of the work with the perforin inhibitor. Graduate student Monica had been working on this project in the basic deletion of different genes from the endothelial wall. That's her work. Phil Swanson, he was the one that showed the smoking gun film of the CD8 T cells sort of attacking the wall itself. Long-standing collaboration with Sue Pierce and her postdoc Brittany Riggle, Jeff Hart and Lou Miller, who's here as well. That wonderful AAV vector came from Jacob in Hamburg. And then that drug, the perforin inhibitor, comes from Joe Trapani in Australia. And that drug essentially blocks the assembly of the perforin molecule. So again, I am finished, and we'll certainly take any questions that you might have if I have any time.
Great talk, thank you. You showed in one of the first images that the CD8 cells go to the cerebrovascular vessels, but not to the skin, I think.
Yeah.
So there must be a difference between the endothelial cells in the cerebrovascular vessel and those in the skin.
Yeah.
What's the difference there?
Yeah, in terms of the, I assume that's what the rest of you need to figure out in terms of what drives the RBC to stick specifically to the cerebrovascular endothelial cell and say not other vascular beds. Things like EPCR and other types of proteins that have been out there in the literature, those are certainly possibilities, but something has to provide that adhesive force that allows the RBC to stick to the wall. And then once that does happen, right, then you have created a situation where any parasite or parasite material that ends up being released from that RBC can be taken up by the endothelium. So infection doesn't have to occur. You just need to have acquisition, at which point the the endothelial cells will cross-present that antigen. But we don't know exactly what the adherence molecule is, again, that drives them to the vessel wall in the brain versus the ear. Yeah.
Thank you, Michael Good from Australia. In cerebral malaria, not all mouse strains get cerebral malaria, and with the same parasite. So is that telling you or suggesting that there's an element of specificity about those CD8 T cells, which is important for the disease? And if so, is there any work to try and identify the antigen that is responsible?
Right. So not all strains drive cerebral malaria, so not all parasite strains drive it, and you don't get it, obviously, in all mice. I think one of the most critical factors is the amount of parasite in circulation. So with some of the strains that we have looked at, where you don't get cerebral malaria, you'll see a parasitemia of about 6-7%, and that typically isn't enough to drive the whole disease itself. You might get micropathologies, but it's tolerated. In terms of this particular strain, the parasitemia will go up 10, 15% in that range. And usually once that happens, you start to get sort of a mass action effect where there's too much vasculature that's being disrupted simultaneously. So I think physical amounts in circulation is a variable. And then the issue or the question that was just raised previously about adherence, right? The degree to which you get physical connections between the RBC in the vessel wall, that's the second variable in this. So either of those things could change the outcome of the disease. Say you have great adherence, but you don't get to the levels of parasitemia needed to drive presentation in lots of different vessels, or you just don't have great attachment. Those are the two things. The peptide, that's certainly possible, that you have one sort of driving immunodominant peptide that is so pathogenic, and that creates the disease. There have been mappings of peptides, at least with Plasmodium strains, but I don't know that that's been looked at as a comparison to a strain that doesn't have it, right? Doesn't have the peptide, doesn't have the strong CD8 response, and then doesn't get the disease. But I certainly think that's another third explanation for how you could get this to happen.
Dorian, that was very interesting. Very important disease. I don't know what the differences are, but certainly the cerebral vascular endothelia do not express aquaporin 1.
So one more time, they do not express.
Aquaporin 1 is present in most vascular endothelia, but not in the brain.
I see.
I don't know what that means.
I don't know if I know what that means. Yeah, I mean, I think in terms of physical movement of water from the lumen into the parenchyma, I don't know that an aquaporin is necessary. I think that even just slight distortions in the tight junction integrity would allow sufficient water to pass through. We see that a lot with immune cells extravasating. So as they extravasate across a barrier, the blood-brain barrier, the barrier opens enough, and you can always see dye, i.e. water, moving across to the abluminal side. And so this attack on the wall may be sufficient, and it doesn't require an aquaporin to generate the water passage. Yeah. Also, the RBC tells you that as well, right? I mean, you have to have a pretty big junctional opening for an RBC to get from this side to the other side, right? That's now a 2 to 3 micron dimension cell to cross.
Thanks again. Great.